Before weighing out the individual doses, the bees must be mixed thoroughly once more. Only then is it possible to guarantee that samples of equal weight contain a nearly equal number of parasites. Over the years, it has been shown that an initial inoculation with about 100 Varroa mites per colony is about right. That number of mites corresponds to a sample weight between 30 to 70 grams of bees, and the weighing must be precise to within 1 gram. Honey jars, covered with fine gauze, are ideal for keeping and transporting the individual samples. The infected bees are now added to the test colonies as an inoculation. In order not to chill, the bees must make immediate contact with those of the winter cluster. As they feel queenless and are well fed, they are accepted without loss. The following year, immediately after the honey harvest, all colonies are treated with an effective varroicide. A new floor inlay must be inserted first. Among others, acaricidal strips are a very effective treatment. They are suspended between frames in the brood nest, and the active ingredient kills all mites on adult bees in a few hours. Any parasites still in cells of sealed brood will be killed only after emergence. A first count is made after 24 hours. This establishes the Varroa population which has been on bees. New floor inlays will reveal the number of mites which had still been in brood cells. Among the debris one finds Varroa mites of various coloration, apart from bees' legs, pupal skins and wax scales. Light-coloured mites are immature and are ignored in this first count. Here the count is done by a computerized apparatus. It reduces the workload when many samples are involved. The samples are prepared by drying and screening. The computer can recognize Varroa mites because of their characteristic size, shape and color. The rate of development of Varroa populations in colonies can vary, and this may depend on a number of innate behavior patterns of the honeybee. One of these is social grooming. When a bee is irritated by a mite, it tries to attract the attention and assistance of neighboring bees by shaking its abdomen violently. This invites them to help with the removal of the parasite. Bees also attempt to shake and brush off the irritating mite, but they rarely succeed because of the instinctive escape mechanisms of the parasite. The effectiveness of social grooming in colonies can be measured by an examination of any mites which had fallen to the floor during periods when no chemical treatment had been applied. Of course, a regular change of floor inlays is absolutely essential for this investigation, and ants too must be prevented from entering the hive. grease-banding all legs of hive stands eliminates that danger. The underside of mites is carefully examined at 40 times magnification. That way one can often find Varroa mites with some damage to their body, here for example on the second and third right legs. The examination must be done on a sample of at least 20 mites, and the percentage of damaged mites serves as a measure of the effectiveness of social grooming in a colony. Another hygienic activity which has an influence on the development of mite populations is the opening and cleaning out of parasitized cells by nurse bees. Somehow they sense a disturbance in the capped cell, but little is known about the signals which trigger cell cleaning activity. The parasitized brood is removed by teamwork of several worker bees. Even though the mother mite usually escapes, her attempt to reproduce has been severely interrupted. The needle test is a rapid and simple method for the investigation of cell cleaning behavior. About 50 cells within a marked area are pierced right to the cell bottom with a fine needle. Colonies with pronounced cell hygiene behavior will have cleaned out most of the pierced cells within 12 to 18 hours. The percentage of cells which had been cleaned out can serve as a numeric value for making comparisons between colonies. All data comprising honey production, gentleness, calm behavior, swarming propensity and Varroa tolerance are assessed at the end of the season. Those stocks which had been in the same testing station can at once be compared, provided the original queens were present at the end of the test period. Under the conditions of fair performance testing, the colonies show their differing qualities. Most characters are distributed normally. This means that very few individuals score very high or very low marks, while most fall into the category of average. Selection means choosing the best individuals for further propagation. The selection difference, the superiority of a character over and above the average, depends on its scatter within the population, as well as on the intensity of selection. Heredity determines to what extent the selection difference is passed on to the next generation as an improvement. With continued selection, the gene pool of a population grows more and more uniform. The possibility of further improvement diminishes with subsequent generations. Yet, the level of performance of a thoroughly selected population establishes itself at a high plane, as long as inbreeding causes no damage. The continuous and collective work of selective breeding has improved the performance and character of our honeybee considerably. The task of selecting for Varroa-tolerant bees confronted breeders with a new challenge. The successes achieved in selective breeding, especially for pronounced gentleness, allowing colonies to be kept in densely populated areas, can only be maintained through ongoing selection.